In this lecture, I want to talk about DNA replication in a little bit of detail, and then discuss some of the mechanisms that the cell uses for repairing various damages and mutations that occur inside of DNA. Now, DNA, of course, is a double-stranded helix, and we know that the bases are what we call complementary to each other. That complementarity of the bases results in the bases essentially holding the DNA double strands together. And the force that makes that possible are hydrogen bonds. We can see the hydrogen bonds that form here on the top between A and T. And we can see that there are 2 hydrogen bonds that hold that together. I think this is something that's pretty common knowledge of students. And that the G's and C's are held together by 3 hydrogen bonds. And the GC hydrogen bonds, of course, being greater than the AT hydrogen bonds, mean that GCs, separating GCs is a harder thing to do than it is to separate ATs. In the process of making DNA polymerase, of course, reads one strand. It sees an A, it puts in a T. It sees a G, it puts in a C, et cetera, et cetera. But one of the things that students commonly forget is what the polymerase is doing besides reading that other strand. And that is that the DNA polymerase, of course, is synthesizing a new strand from that. So that synthesis involves more than just A for, on the other side of a T. It also involves the formation of phosphodiester bonds. So phosphodiester bonds are these bonds that you see here between adjacent sugars. So when we look at the strand of DNA, we basically see a nucleotide linked to a nucleotide linked to a nucleotide. And we remember, of course, that a nucleotide contains a base, which is up over here. It contains a sugar, and it contains at least one phosphate. In the case of a strand of DNA, there is only one phosphate. We start with a triphosphate in the synthesis that we can see down over here, and we end up after the synthesis with a single phosphate here. The phosphodiester bond arises from the fact that this phosphate is linked to the sugar above it and to the sugar below it. It's linked to the carbon number 3, the hydroxyl was on carbon number 3 on the top, to the hydroxyl was on carbon number 5 on the bottom. So that's what makes a phosphodiester bond. So it's important to recognize that phosphodiester bonds are the forces, the covalent forces that hold together the individual nucleotides within a strand of DNA. Now, the synthesis of that strand of DNA turns out to be fairly complicated at the protein/enzymatic level. We look at a replication or replicating DNA, and on the surface it appears to be quite straightforward. We see, for example, here replication occurring within a replication fork. And I've talked in class about how the replication proceeds by 2 mechanisms. One mechanism that I call the leading strand replication, and one strand that is the other strand of the duplex that is synthesized by what's called the lagging strand mechanism. And this is a point that students commonly find confusing. They don't understand why, for example, there's a leading strand synthesis and there's a lagging strand synthesis. So what I want to do is spend a little bit of time discussing why that occurs. So if there's a couple things to remember when we think about the replication of DNA. First of all, of course, the polymerase is looking for the right complementary bases to put together. Second, the polymerase is making phosphodiester bonds. And the third is that, of course, all DNA polymerases need a primer. I've discussed that in class. And that primer in the cell is comprised of RNA that's made by an enzyme called primase. And I'll show a figure for that in just a little bit. So let's look to see what happens then in the synthesis of the leading strand. If we look at the leading strand, we see at the 5' end, there is that little RNA primer. And at the end, at the 3' end of that RNA primer, the DNA polymerase has started synthesis, and it's made this strand of DNA going along here. The last thing we have to remember with DNA replication, and this is really, really critical, is that DNA polymerization only occurs in the 5' to 3' direction. And since the strands are anti-parallel, we see the synthesis going this way. That is, it's going opposite the polarity of the strand it's being copied. So the strand that's being copied in the same orientation is in the 3' to 5' direction. That's how we get strands that are anti-parallel to each other, like we see right here. Well, that's fine for the leading strand, because in the leading strand, all that has to happen is that the polymerase just keeps moving forward, 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 and as a consequence, this top strand gets unwound from the bottom strand, and it just keeps going further along the way into here. The bottom strand replication, however, is considerably more complicated. And the complication arises from the fact that, first of all, there has to be a primer, and second of all, the replication can only proceed in the 5' to 3' direction. So we actually see that down here. If we look, for example, we say, well, okay, look at the bottom strand. The bottom strand that's being copied goes 5' to 3' that way. So that means that replication of that strand can only go 5' to 3' from left to right, or down as I'm showing in this case. Now, why does it happen in pieces? Well, remember that the primase can only copy a single strand after it's been unwound. And that strand gets unwound only after this strand moves further along. So as this strand keeps moving further and further along, remember I said the top strand gets unwound from the bottom strand, that opens up a region of the bottom strand that can now be replicated. So the primase comes along and says, oh, look, I can put a primer down here, it puts a primer down, and then DNA polymerase comes along and fills in until it gets up to the point of where the last one was. So this goes on over and over and over and over during the replication of a double helix. And it's for this reason that we see that the leading strand replication occurs in one piece and the lagging strand replication occurs in the bottom piece. The lagging strand replication occurs in multiple pieces on the bottom strand as shown here. Now, if we look at the proteins involved, I've actually simplified it on the last strand. There's a lot more complexity to what's there. I've talked about these in class a little bit, but I want to just go through these proteins again here so that you have an understanding of what's there. First of all, the DNA polymerase, well, we're looking at a replication fork. The replication fork that we saw in the last strand in the last slide was laid on its side. In this case, we've taken that and we've stood it up so that the forks of the replication that are occurring are shown down here, okay? And the leading strand synthesis that's occurring is starting here at the bottom and it's moving upwards. So we see leading strand synthesis occurring here. It's occurring in one piece and it's moving upwards in the direction of that fork that we saw on the last slide. The lagging strand synthesis, on the other hand, is occurring over here on this strand, and it's occurring in pieces, and you can see a couple of those pieces as we look down through this region here. Now, there's more involved in this replication than the DNA polymerase, and so that's what I want to spend some time talking about in here. If we look here, we see this double-headed thing here. This double-headed thing is DNA polymerase 3. And DNA polymerase 3 is a duplex, meaning it's two identical copies of a protein that are there. One part of the duplex is making the leading strand, and the other part is making the lagging strand. Now, these two can actually occur simultaneously in the cell, except for remember, of course, that the lagging strand is made in pieces that we will see. If we look at this, we see that there's actually a looping that happens in this process, and though that's not really critical for you to know, I show it because there actually is a loop that happens because the rate of synthesis of the leading strand is different than the rate of synthesis of the lagging strand. So if the leading strand gets way ahead, the lagging strand has a big single-stranded piece that can just hang out there, and that's what actually makes the loop. It doesn't matter for our purposes. We can see on this slide some of the players in the overall process. We can see, for example, the primase that makes the RNA primers that are necessary to start the synthesis. And we see a couple of other proteins that I haven't described, at least in this talk, in some detail. The first of these proteins is a very interesting enzyme known as helicase. And so helicase is critical for DNA replication in E. coli to occur rapidly and efficiently. Helicase's function is to use the energy of ATP to basically unwind the strands. So this enzyme right here is peeling apart those strands. And so remember, during the leading strand synthesis, I said that the top strand in the last slide had to be unwound from the bottom strand. That's the job of helicase. Helicase is unwinding those strands, and it's unwinding them, as I noted in class, at the rate of 6,000 RPM, an incredible process, because remember that in E. coli, this replication process, this whole complicated scheme that's occurring here, this replication process is occurring at 1,000 nucleotides a second. It's just an incredible thing. So helicase's function is to separate the strands. An enzyme shown immediately above the helicase also is important in the replication, and it's known as DNA gyrase. And this figure actually exaggerates or brings them closer together than they literally would be in the cell, because there needs to be a bit of a gap between them. You'll see why that's the case in just a second. Imagine if helicase is ripping apart these strands and nothing else were to happen. If nothing else were to happen, we would see unwinding of the strands here, but by unwinding strands here, we would be creating tension ahead of where that DNA gyrase is, so that tension will result in essentially tangling up of the DNA. And if we don't untangle that DNA, the DNA will become a knot, essentially, and replication would cease, the DNA might break, et cetera, et cetera. So the cell has to have something that relieves the tension that's created by the helicase. The helicase is creating a tension in that DNA duplex ahead of the replication fork. So the gyrase is literally a little further up a ways here, where it's on the other side of where that tension is. And the job of DNA gyrase is to make cuts in the DNA that relieve the tension, and then it puts the cuts back together. That is, it breaks phosphodiester bonds, allows the tension to come out so the DNA relaxes a bit, and then the DNA gyrase remakes those phosphodiester bonds to join the strands back together. So DNA gyrase is an example of an enzyme that we call a topoisomerase. And so I will use either term there. DNA gyrase is a specific type of topoisomerase, whereas there are other topoisomerases that are present in cells that do other things. So for our purposes, we will think of topoisomerase and DNA gyrase sort of interchangeably, but suffice it to say that DNA gyrase is a specific type of topoisomerase. There are some other players that are important in the replication fork, which is what we're seeing here, that allow the cell to efficiently go about replicating DNA. One of these players is a protein known as single-stranded binding protein, or as it's labeled on here, SSB. SSB has the important function of protecting single strands, and it protects them simply by binding to them. Now, protecting single strands is important because damage to a single strand, let's say a cut that would happen, would essentially destroy the replication fork. So putting a protein on there to protect that strand when it's in the single-stranded form is important. If I were to make a cut in one of the strands of a DNA duplex, the consequences would not be so large because an enzyme like DNA ligase, for example, could in fact repair that and everything would be fine. But if I get a cut in a single strand, there's no other strand to help hold it together and everything would fall apart as a consequence of that. So single-stranded binding protein, or SSB, is important in protecting that. SSB also appears to play a role in helping the replication to proceed efficiently. There are two other enzymes that are important in the replication of DNA. Remember that we had in this initial synthesis of each of the strands or pieces of the DNA, that is the leading strand or the pieces on the lagging strand. At the beginning of each of those strands, there was a little short fragment of RNA. And that short fragment of RNA is problematic because it has to be removed. If we're going to have a duplex of DNA, we don't want to have RNA in it, because what happens is if the RNA were left in there, when the next DNA replication came along, the DNA polymerase would hit that RNA and it would stop because most DNA polymerases will not copy RNA. So that means that the RNA has got to be removed during or after the primer has been made. It turns out that the removal of that primer is the function or partial function of another enzyme that makes DNA in the E. coli cell. And the removal of that primer is the function of an enzyme known as DNA polymerase 1. Now remember that DNA polymerase 3 is the main replication enzyme. It's making the leading strand, it's making part of the lagging strand, the part after the primer, and it's moving along this thing at 1,000 nucleotides a second. So it's really, really efficient, okay? The primers have to be removed, and they're not so rapidly removed as this guy is moving forward. DNA polymerase 1 is responsible for that. So how does it do what it does? Well, when the fragments are made, you notice on the figure that I showed that there's a gap between each fragment. And so that gap is a binding site for DNA polymerase 1. It gets on the 3' end of each of those fragments, and that 3' end is a DNA end. So on the back side of it, we can think of where it's bound to that fragment. The back side is DNA, and as it looks forward, what does it see? It sees the 5' end of that primer. Well, what DNA polymerase 1 does is it starts filling in that gap. It starts putting in an A for a T, across from a T, a G across from a C, et cetera, et cetera. And it gets up there and it hits that primer. And when it hits the primer, DNA polymerase 1, and only DNA polymerase 1 in E. coli, has the ability to chew up that RNA primer. That ability to chew up that primer is part of another enzymatic activity that's found in DNA polymerase 1, and that activity is known as an exonuclease. It's specifically an exonuclease that works in the 5' to 3' direction. Now I want you to pay attention to that. There are other exonuclease activities associated with DNA polymerases that I'll talk about in just a minute. But this activity, the 5' to 3' exonuclease activity, is unique to DNA polymerase 1. And that exonuclease activity, the 5' to 3' exonuclease activity of DNA polymerase 1, is what removes that RNA primer. Now, it's pretty cool what it does. As it removes the primer on the front side, on the back side it's filling it in with deoxyribonucleotides. So in essence, the RNA is converted to DNA in the process. When the polymerase is done, it leaves a tiny gap between the last nucleotide it put in and the first nucleotide in front of it. That phosphodiester bond between those two nucleotides is not closed by DNA polymerase 1. It has to be closed by something else, and the something else that closes it or links those together is DNA ligase. DNA ligase closes the very last gap. So DNA ligase is important for linking together all of the Okazaki fragments, that is, those fragments of the lagging strand, okay? It's responsible for linking together all of those fragments into a contiguous piece. After DNA ligase has done its job, this strand on the leading strand is in one piece, and all of these individual pieces of the lagging strand have been joined together into one piece as well. At that point, the replication process is essentially done. Now, there's only one other protein on this figure that I haven't described, and I want to say a word about it. It is not necessary for the actual replication, but it's necessary for an important property of the DNA polymerase 3. And that's known, it's a protein known as the beta clamp. And so the beta clamp has a very simple function. It binds to the DNA polymerase 3, meaning it just holds on to it, and the beta clamp has a structure that looks like this. So we can think about the clamp as looking like my hand, and it exists in two forms. One, the open form that we can see here, and the other form closed like this. So what happens is that the beta clamp, when it is open, it can grab a hold of a strand, and then when it closes, now it has wrapped around that strand. Okay? Well, it's not held tight, that is, it can slide along, but what it allows the polymerase to do is not fall off. So DNA polymerase III has a very important property. Once it gets onto a DNA, it rarely comes off. And the only time it comes off is when the beta clamp, after it has finished its thing, opens up, and then everything can come off. DNA polymerase III binds to the beta clamp and gives it that property. DNA polymerase I, down over here on the other hand, does not bind to the beta clamp. So because it doesn't bind to the beta clamp, DNA polymerase I has this property. It will go onto a DNA for a while, and then it will fall off. And that turns out to be a very useful property if the fragments that the polymerase is making are scattered, and the polymerase I doesn't need to stay on there, because remember, it's only synthesizing DNA for that very short segment of where the primer was. So the beta clamp allows the polymerase III to stay on, and it doesn't affect the DNA polymerase I.